All right, so the idea of this activity is twofold, okay? One, to kind of review the factors that can affect the growth of a plant, right? We talked about some of those things like light and soil and water and all those kind of things that a plant requires, okay? But also to review the scientific method, okay? Specifically experimental design. All right, so we want to review kind of how we build a proper experiment, things like that. We did that very first lab of the year, okay, where you had to design the procedure, okay? Same idea here. So you're going to be completely designing a lab experiment to study the effect of a variable of your choice on the growth of plants. So it can be anything. Okay, you can choose any variable you want that could affect the growth of plants. Okay, I've seen everything from as mundane as I'm going to give them different amounts of water, which is not very creative, okay, to I had one person who was going to have a bunch of different plants that all got different colors of light to see if that affected their rate of photosynthesis and their growth, etc. Okay, so it's entirely up to you what you test, as long as you test it using a controlled experiment. How many manipulated variables can a controlled experiment have? One. Okay. If you have more than one manipulated variable, intentionally or otherwise, you will never know which one causes the change in your experiment. Right? So when you set up your experiment, you have to be very careful that you only actually have one manipulated variable. Okay? I very rarely have anyone say, I'm going to manipulate these two things, but they say, I'm manipulating this, and then their procedure doesn't take into account some other things, and they end up having more than one. Right? So an example could be that I had a person who said they were going to test the effect of different temperatures on the growth of a plant. So they put one plant in the sun and another in the fridge. What happens to the light in the fridge when you close the door? It goes off. So now they unintentionally are testing, I've put a plant in the cold dark and another one in the hot sun. Okay, so you have to make sure that you're keeping everything else that could affect the growth of a plant the same, except for the one thing you're testing. All right, everyone kind of follow me there. All right, now your manipulated variable can have multiple forms. Okay, I did once have a person who did designed theirs, and they tested different soil types. The manipulated variable was the type of soil, but they tested regular potting soil, rocks, sand, clay. Okay, they had all these different soil types. Yes, it looks like there's more than one manipulated variable, but there really isn't. They are testing the effect of different soils, plural. Okay, still only one manipulated variable. Everyone kind of follow my reasoning there? Okay, just make sure you're only testing one thing. Okay, had they tested, different soil types and planted a potato, a carrot, a bean, and a turnip, okay, now it's not a controlled experiment because they're not all the same kind of plant. All right, so make sure you're testing, you're keeping everything the same except the one thing you're trying to change. All right, so like we said, that variable could be nutritive, so the amount of water, sunlight, fertilizer, whatever. Okay, it can be conditional, where the plant is going to be, temperature, or whatever. Okay, the first thing that should appear in this experiment is an abstract. That's not something that we've had in an experiment before. All it is a short paragraph that explains what you know about the subject you're testing. All right, so you could say, like if you're testing the amount of light that a plant gets, you could say light is important for plants because they carry out photosynthesis, and light is the source of the energy that does that, blah, blah, blah. You have some little blurb about why light is important. Okay, that would be your abstract. Okay. Then after that, it sets up just like any other lab. Your problem, your variables. So problem, design, and then hypothesis, materials, procedure. Okay, it should look just like, and I think the template is there. I've got all those things in there for you. You just have to fill them in. Right? So this time you're actually making up the problem. So you have to say, the problem here is what is the effect of blah, blah, blah on the growth of a plant. Okay, so identify and explain your variables. This is the manipulated variable. This is why it's the manipulated variable, how it's being manipulated. This is the responding variable. This is how it will respond. What is everybody's responding variable going to be here? What specifically about it? Its growth. Now, when you're describing its growth, should you also make mention of how you're going to measure that? Yeah, because there's a lot of different ways to measure the growth of a plant. It may have to do with the type of plant you're planting. Okay, if we're talking about a carrot, I mean, you sure might want to measure how tall the foliage above ground gets, but you might also want to measure how deep the carrot gets, or how big or how heavy the carrot gets, or something like that. So make sure you list in there what kinds of things you're going to use as your measuring stick for growth. You're going to count the number of leaves, or how much fruit this thing makes, or whatever. Okay. And then your controlled variables, you're going to need 3 of them for sure. Okay, make sure that you're controlling all of the things that could affect the growth of a plant other than what you're testing. And you're going to want to really address that in the procedure, right? So you're going to need a list of materials. Okay, so in the materials, you would identify like what kind of plant you're using, what kind of soil you're using. Are you just going to Home Depot and buying a bag of potting soil, or are you using something else? Okay, how much water? Okay, are you going to need water? Okay, all of that kind of stuff. Put your list of materials. All right, your procedure needs to be detailed. It needs to be more than plant the seed, water it, and then measure it. Okay, if that's all you have for your procedure, it's not going to go very well for you. Okay, what I'm looking for in terms of detail in the procedure is things like, how much water are you giving it? How often are you giving it that water? Are you giving it anything else? And how much of that are you giving it? How often are you measuring it? How much light is it getting? Okay, all those kind of things. All those things need to be mentioned in the procedure so that someone could replicate your research later. So you do need to make mention of all the little mundane details that are part of running a proper experiment. Everyone kind of follow me on that? Okay. That's it. Once you're done the procedure, that's it. You don't have any data because you're not actually running it. Without data, it's impossible to do analysis. Without analysis, there can be no conclusion. So you're only doing the abstract problem, hypothesis, abstract problem, design, hypothesis, materials, procedure. First half. Okay, you don't have to do the rest of it. Okay, and you should be able to easily get that done in class today. Right. So a variable of your choice, how it would affect the growth of a plant, and experiment to test it. Questions before you get started? Okay, if there are questions while you're doing this, just let me know. Okay? All right, have at her.